we've talked about DNA, its general structure, how it replicates, and RNA. Now let's talk about where it is all useful. So if we look at our prior knowledge, in the top box here, we see that we have BB. Those represent two alleles. And in this case, those alleles represent having blue eyes. Well, we know those alleles are variations of a gene. And a gene is located on a chromosome. And the chromosome is made up of DNA, which is in this tightly coiled double helix. That's what you see in the second box. And then we have that DNA is going to code for those genes and give us the traits. But how do we get there? So if we think of moving from genotype to phenotype, remember genotype is going to be the actual genetic makeup, the alleles and genes that you have that give you your appearance, and the phenotype is that appearance. It's what you look like. So if we look at this, we see DNA is going to be transcribed or go through the process of transcription to become RNA, and then RNA is going to go through a process called translation to become a protein. And let's look at those a little bit closer now. So transcription is going to be the process of using DNA to make a copy of the mRNA. And we can look at an animation.
The two strands of the DNA double helix are held together by bonds between complementary base pairs. DNA encodes genetic information in the sequence of bases. The portion of the DNA molecule that is a single gene or coding region is bound at termination and promoter sites. A molecule of RNA polymerase binds to the promoter site. It moves along the DNA, separating the two strands of the double helix. Each now unpaired base will bind to a nucleotide in the vicinity which has the appropriate complementary base. In the synthesis of RNA, uracil is the nucleotide complementary to adenine. This process ceases when RNA polymerase reaches the termination site. The DNA strands bind to one another once again as the new RNA molecule moves away. This RNA is a copy or transcript of the message contained in the gene.
Going back to our notes, now that we've seen what transcription is, it is that making a copy of the DNA. We can look at how that's done. So same enzymes that helicase is going to act as a scissors and cut those bonds apart between the DNA strands. And then we are going to match up the complementary base pairs. But remember that in RNA, it's uracil instead of thymine that's going to bind with the adenine. So to make a strand of mRNA, that's the first RNA that's going to be transcribed, we're going to look at the strand of DNA, and this is happening in the nucleus. We can see that T is going to match with U, A to, T to A to U, C to G. And we're going to keep going on. T to A to U, G to C, G to C, T to A, C to G, C to G, A to U, T to A to U, T to A, and C to G. Now, we have our mRNA strand. And you can see that these are split up into 3 bases at a time. And we call that a codon. So each triplet on this strand of mRNA is going to be called a codon. And that's going to code eventually for an amino acid. So we also mentioned in the little video promoter sites and termination spots. There's going to be certain codes, like this AUG, which actually eventually you'll learn is a code from thymine, is going to be a start codon. It's going to tell, um, it's going to send the message to start coding at this point and start reading it. Then once the mRNA travels out of the nucleus, it's going to go to the ribosome and it's going to be translated. Now, translating is a process of taking mRNA and decoding it to make an amino acid sequence. Let's look at a translation animation.
A transfer RNA molecule is composed of 5 nucleotides. Each transfer RNA molecule recognizes only one amino acid, which becomes bonded to the top of the transfer RNA molecule. Located on the bottom of the transfer RNA molecule are 3 nitrogen bases called an anticodon that pair up with messenger RNA codons during translation. As translation begins, the first codon of the messenger RNA strand attaches to a ribosome. Then, transfer RNA molecules, each carrying a specific amino acid, approach the ribosome. When the transfer RNA anticodon pairs with the messenger RNA codon, the 2 molecules join. Often, the first codon for messenger RNA is AUG, which codes for the amino acid methionine. AUG signals the start of protein synthesis. The messenger RNA then slides along the ribosome to the next codon, where a new transfer RNA molecule carrying an amino acid will pair with the messenger RNA codon. The 2 adjoining amino acids then become joined by a peptide bond, and the first transfer RNA molecule is released by the messenger RNA. As the process continues, a chain of amino acids is formed until the ribosome reaches a stop codon on the messenger RNA strand.
So what we saw was translation, and that's when an anticodon, which is attached to an amino acid, is matched up to the codon on the mRNA. So the anticodon is attached to the tRNA, which is represented by these little structures here, and the 3 bases are going to be the anticodon, which are going to match or be the complements to the codon on the mRNA. And then on that tRNA, we're going to have the amino acid. Eventually, those amino acids are going to be joined by peptide bonds, which are going to make a big chain of amino acids or peptides, and it will become a complex protein. Now, to figure out what each of these codons codes for, we have this little genetic code chart. And it looks kind of funny, but it's pretty easy to read if you start from the center. So to determine the amino acid, you're going to start at the middle, okay, with these 4 letters, and then you're going to work your way outwards. So, for example, if we're looking at UCG, CAC, GGU, we break it up into the codon pairs. We would see that UCG, okay, if we start with U, C, and then go to G, we get serine. Then CAC, start at C, and do this with me. The next one we go to is A, and then we find C. So we're going to get histidine. And then the third one, we start at G, and then we go to another G. So from G to G, and then a U. So we get glycine. And that's how we would figure out what the code is actually saying by those letters. If we do some practice with translation and we have a code, we'll start with the code that we started with. So we started with AUG, AUC, CAG, GUA, and UAG. Okay, now this is at the ribosome. The anticodon is going to be the complement of that. So it's going to be UAC, UAG, and then GUC and CAU. The structure here, right, is this whole thing is the tRNA, and that tRNA is going to be attached to an amino acid. So here, if we looked at our chart, we would find methionine. Let's go back to that chart actually and find the next one, which is UAG. So let's find U, A, and then G. Okay. Is actually a stop codon, which doesn't work. Um, oh, that's because we need to do AUC. So if we do A, U, and C, we get isoleucine, which we're going to abbreviate as I-Lu. And then let's do this one. We have GUC. So GUC, all right, if we were to go to G, U, and C, we'd get valine. But like I did before, it was actually a mistake, and that's what I wanted to show you. Because we came up with the stop codon before, then you know something's wrong. So this example, you don't realize that something's wrong, but you can check yourself with that if you come up with a stopper, in the middle of a sequence. What you should be doing is looking at the mRNA strand to figure out what amino acid you're coding for. So in this case, the mRNA would be CAG. So it would be C, A, and then G. So we would get glutamine. So glutamine will go like this. And then the last one would be attached to CAU, but it's UAG. So again, we're going to go U, A, and then G. And that makes sense to have a stop at that point. Which is actually valine. Okay. So this whole thing here, this structure that we have, all those amino acids are going to be joined by peptide bonds, and that forms our polypeptide or our protein. That's the process of translation and transcription.